We all know about viruses and bacteria as pathogens, agents that cause disease. But how about prions? Prion diseases, also known as transmissible spongiform encephalopathies, or TSEs, are rare neurodegenerative diseases that affect humans as well as animals. They have a long incubation period and are universally fatal. Death usually occurs within several years due to the rapid progression of the disease. There are currently no cures or vaccines. In humans, symptoms usually include a decline in intellectual function and a gradual loss of physical coordination. One example of a human prion disease is Creutzfeldt-Jakob disease. One well-known example of an animal prion disease is bovine spongiform encephalopathy, commonly known as mad cow disease. Prions are essentially misfolded proteins. All proteins have a specific structure that enables them to carry out their function. This structure involves a specific sequence of amino acids, with different sections called into alpha helices, folded into zigzagging beta pleated sheets, or keeping an irregular structure. The final 3D structure is maintained by interactions between different areas of the chain. Changes to this structure may interfere with the protein's ability to carry out its function. With prions, this change in structure makes the protein infectious. Prions are able to convert a specific type of protein, known as prion proteins, from the normal form to an infectious misfolded form through contact. The normal form of the prion protein is rich in alpha helices, while the misfolded form is rich in beta pleated sheets. These prions aggregate into insoluble threads of protein known as fibrils, which then accumulate to form plaques. Prion proteins are found most abundantly in the brain, so the brain and the rest of the nervous system are hit the hardest by the disease. Also, prions are resistant to the normal processes of protein destruction and replacement. Normal proteins are regularly destroyed by the cell using proteases and replaced with new proteins. Prions, however, are resistant to proteases because of their very stable structure, so they are not destroyed. Instead, they accumulate exponentially by continually converting normal proteins into the abnormal form. These clumps of prions damage or destroy neurons, causing the formation of tiny holes called vacuoles in the brain. This gives the brain a spongy appearance, giving rise to the term spongiform encephalopathy. Symptoms vary depending on which part of the brain the prions accumulate in. For example, if prions accumulate in the part of the brain that controls vision, the patient might develop visual problems. Prion diseases develop in 3 ways. Firstly, they may be hereditary. If there is a mutation in the gene that codes for the normal protein, there is a higher chance that the protein will misfold itself and become a prion. Secondly, they may be sporadic, meaning that the disease appears spontaneously for no known reason. Thirdly, they may be acquired, meaning that the patient was infected by prions from an external source. This may occur through oral transmission, for example by eating food that has been contaminated with prions. Kuru, a type of prion disease, was transmitted between the 4 people of Papua New Guinea through ritual cannibalism. 5 young people died of CJD between 1998 and 2000 after eating meat from 2 butcher shops where the butchers, following traditional methods of slaughter, had opened the animal skulls and removed their brains themselves. They then used the same tools, now contaminated with prions, to butcher the meat that the patients later ate. Prions may also be transmitted through medical procedures such as corneal transplants and pituitary hormone injections, or through the use of contaminated medical equipment in neurosurgery. Prions, unlike bacteria and viruses, do not contain nucleic acids, so they are not destroyed by standard sterilization procedures such as heat, alcohol, formaldehyde, and radiation, which target and destroy the nucleic acids that direct replication in viruses and bacteria. Medical equipment that has been used on medium to high infectivity tissues, such as brain tissue in patients with prion disease, is not reused. Alternatively, the equipment may be subjected to decontamination procedures that involve protein hydrolysis or denaturation, for example by using bleach. There are currently no cures or vaccines for prion diseases, and a lot is still unknown about the infectious agents themselves. Current research on prions focuses on the function of the normal prion protein, the development of new diagnostic methods such as blood tests, other methods of transmission, and potential human exposure to prion diseases. Rapid developments are continually being made in this field, and it seems that it will remain highly exciting for the foreseeable future.